Hi. Eukaryotic genome is much more complex in terms, not just in terms of the informational complexity, but also in structural complexity, meaning that it is bound around nucleosomes or bound around histones to form nucleosomes and then further compaction into chromatin. And also the spaces between the genomes, that is the intergenic space, is huge, and at the same time also within the genes, that is in the form of introns, the space is huge. That means transcription of the eukaryotic genes is going to be complex because the chromatin that is highly compacted should be opened up and then transcription should have happened. That is what we will try to discuss right now. Eukaryotic transcription. The primary differences between prokaryotic and eukaryotic transcription in terms of RNA polymerases is that eukaryotic polymerases have something like 3 different RNA polymerases. So we can, and then the why do we need 3 is something peculiar in terms that RNA polymerase 1 is for rRNAs, and the RNA polymerase 2 is the one that is major RNA polymerase that transcribes majority of the mRNAs and also small nuclear RNAs, and RNase polymerase 3 is for transcribing tRNA genes and also the small RNAs. So to simply remember, RNase polymerase 1, Pol 1 for RNAs, Pol 2 for mRNAs, Pol 3 for tRNAs. Of course, there are small additions such as small nuclear RNAs for RNA polymerase 2 and small RNAs for RNA polymerase 3. Apart from that, there are a huge number of general transcription factors which are involved and involved in the transcriptional initiation and also elongation. So because there are 3 different RNA polymerases, we can also expect that there are 3 different types of promoters as well. And majority of the recognition, we also need the accessory proteins, and even more important are something to open up the chromatin so that the promoter becomes accessible, and so on, are regulated by a huge complex called as mediator complex in vivo at least. And binding, there are several other proteins such as chromatin modifying enzymes. There are, by way of changing the histone tail modifications such as methylation, acetylation, the chromatin can either be loosened up or compacted together, highly compacted. So these proteins are also important, those containing chromodomain and bromodomain, and many other regulatory proteins as well. So eukaryotic transcription as such is a complex endeavor in terms of regulation structurally and also that of informationally. So we'll start with some of the essential features or brief core essentials of each of these promoters. Type 2, the polymerase, the best understood one is of RNA polymerase 2, or best studied at least, and the core promoter consists of about 40 to 60 base pairs, and these are something like this here, and the one broken arrow here is the +1, which is the place where the first base is added. From here on, the transcription starts. So this would be the downstream and this side would be the upstream. What we have here is a Tata box which is rich in TA regions. Because it's TA regions, there are likely less number of hydrogen bonds, so it will be it is good for easy for melting to occur. And there is also another region called as BRE, which stands for transcription factor 2B recognition element. Say here transcription factor 2B recognizes BRE. TF stands for transcription and 2 stands for the element that is the promoter that is it is specific for. Here we are talking about core 2 promoter, so that is what is the Roman number 2 stands for. Tata box is bound by a protein called as Tata binding protein. And then we have the initiator sequence where the actual denaturation and actual polymerization occurs. And then there are also several other elements, promoter elements called as downstream core element 1, 2 and 3. And there are also downstream promoter elements like these. So these regions are bound by the protein TF2D, and not that all promoters of RNA polymerase 2 will have all of these. Some may be missing in one or more, but at the same time there are also many other factors that are involved in the regulation or even in the expression of these genes, many of the mRNA encoding genes. So that's about core 2 promoter. Similarly, we have for core 1 promoter, that is, it is much more simpler in terms that way. This is the core promoter that is present, which is where the transcription would start, that's +1. And it is about some 60 bases in length. And we also have UCE element, that is upstream control element, that is where the protein called as UCE binding factor is bound. UBF, and we also have another protein called as SL1, which also has several domains, which is kind of putting it together several transcription factors together, including TBP. So SL1 will only bind if UBF is bound to the UCE, and so that is when these two together will recruit RNA polymerase 1, and then transcription of these genes would happen. Transcription of rRNA genes, because this is promoter for 1, which is RNA polymerase 1. So majority of the rRNA genes would have this type of promoter organization. In the RNA polymerase 3, they recognizes promoter, which is a little bit different in the sense that the promoter elements, or majority of them, are in downstream, that is box A and box B, and that is recognized by transcription factor 3. We are calling it 3 because this is related to RNA polymerase 3. Transcription factor 3C, here we have transcription factor 3B, along with the Tata box binding protein domain or protein. As a complex, it would bind and transcribe majority of the tRNA genes. So tRNA genes and all have many of these this kind of promoter with box A and box B. If you remember, RNA polymerase 3 also transcribes 5S rRNA. So there are only subtle differences about what else is involved. Say TF3B and 3C are involved in the expression of tRNA genes, whereas 3B, 3C and 3A are involved in the expression of 5S rRNA genes. So this is how the organization or the promoter elements and the transcription factors are involved in the transcriptional initiation of in eukaryotes. So we have to remember that there are 3 different RNA polymerases, 1, 2 and 3, and they have different types of RNA. The promoters themselves also differ, and there are different types of transcription factors associated with initiating transcription from the corresponding promoters. Thank you very much. We'll see you again.